Welcome everybody to our webinar. My name is Oliver Rinner. I'm CEO of Biognosis, and today I'm happy to introduce Andreas Beyer from the University of Cologne. So Andreas is a true systems biologist who integrates in a quantitative way highly dimensional protein, RNA, genetic and interactome data. So he studied systems science at the University of Osnabrück, held postdoctoral positions at the Leibniz Institute for Age Research in Jena and at the University of California in San Diego, in the group of Troy Heidegger. From 2007 till 2012, he was an independent group leader at the Technical University of Dresden. So today he's a professor for systems biology at the University of Cologne. Now, before I hand over to Andreas, let me give you a short introduction to Biognosis. So our mission is really to enable researchers to better understand their biology by enabling them to analyze the proteome. And we do this in two ways. For people that don't have access to mass spectrometers or not proteomics scientists, we provide contract research services, for instance, in pharma, biotech, but also academia, where people send us their samples and we perform projects for them. On the other side, for the experts that do have access to mass spectrometers, we provide software and reagents so that they can do next generation proteomics themselves. So Biognosis has its roots in the group of Rudi Ebersold, ETH Zurich, where almost 10 years ago we spun off the company. And since then we invent, develop, and provide cutting-edge proteomics to the R&D market. We are still close to Zurich with our state-of-the-art facilities here, and also have a site in Beverly in the USA. Now, at its roots, Biognosis is a science company, but from the beginning we were working with customers globally from pharma, biotech, and academia as we developed the company. And it is actually the reason why we asked Andreas today to talk about the relationship between proteomics and transcriptomics, that many of our customers ask us about this. So we are interested in this very much ourselves, but our customers are as well. And as a proteomics company, we are certainly focused on providing deep and quantitative protein expression data. So this is what we develop and what our ambition is. But obviously, many of our customers that are doing discovery projects, for instance, are using a wide array of methods, including RNA expression data, certainly. And I think we all certainly know that RNA expression and protein expression are not the same. And I think we all feel that there's something to gain if we would combine in the right way RNA and protein expression data. But the hard question is how to do this. So I'm very much looking forward to hear more about this from an expert here in this webinar. We will have a Q&A session in the end, and we collect the questions that come in. So you should see this question tab in the tool. You can type up your question anytime that comes to your mind. We will collect it and have it here. In the end, in the Q&A session, we will address it. And now I want to hand over to Andreas, giving him the control over the mouse. All right.
Okay, yeah, this seems to work. Okay, yeah, thank you, Oliver, for the introduction, and also thank you for inviting me to doing this webinar, because this is a topic that I find really fascinating and interesting, because there's no simple answer to the question, yeah, to what extent or how much the protein concentrations depend on mRNAs. This is being researched for decades, and it's very complex, and I like complex problems, or problems that are not so simple. But of course, I'm going to try, at least today, to make it as simple as possible for you, and to somehow reduce the complexity if possible. So we go to the first slide, which shows the outline of what I've collected for today. First, I'm going to talk about protein mRNA correlations. So you simply plot protein concentrations as a function of the concentrations of their coding mRNAs, and what is this going to tell you? And what are the pitfalls there, because there are quite a few issues. Then the next section is a bit more about the biology, so what really determines protein levels, and what determines what we actually measure in the mass spec. And then when is it important to look beyond the transcripts? When is it necessary to look at post-transcriptional regulation? And here's a disclaimer. Post-transcriptional regulation includes, of course, also post-transcriptional regulation of transcript levels. There are other mechanisms that change transcript levels beyond transcription, of course. I'm not going to talk about this. So what I'm really going to talk about is, given you already know the transcript concentration, what are the additional processes on top of that that change protein levels? And then the last very short section is on what else can you do with protein mRNA correlations other than just investigating the biology. So here I'm showing you a very simple examples how you can actually use these types of correlations also for quality control. Now, of course, because the time is limited, I can only very briefly touch on some of the aspects in each of these chapters, and everything is very incomplete, and there are many other things that are interesting and important. So actually, most of what I'm going to talk about is based on a review that we published last year in Cell. So there's much more detail in this review. Also, if, I don't know, there are aspects session at the end, you can the presentation. Now the mouse is, now we go. Okay, so first question, protein mRNA correlations. What are these correlations and what are they telling you and what are the problems here? So if you plot protein concentrations as a function of mRNA concentrations, as it's shown here in this dummy plot, you actually get pretty rich information. So first of all, it of course shows you how much the protein levels depend on the mRNA levels. So you can quantify the contribution of the mRNA to the protein level. But we and others have also looked at the deviations from this regression line, and we think this also tells you something about these post-transcriptional processes that I'm also going to talk about later. So how much does, for instance, translation, variation in translation, or variation in protein stability also influence the protein levels on top of the mRNA? But of course, and this is also important to keep in mind, the deviation from this perfect regression line is also always a subject to noise, and there could be also measurement errors that, of course, don't give you a perfect correlation. But I think there are a number of publications that have shown already that this is very interesting, also information-rich to look at these residuals, the deviations from the regression. But you find this in this cited in the review. Now, this is very important. There are different types of these correlation analysis that you can do, and it's very important to distinguish them. And this is what I'm trying to do on this next slide. So on the left-hand side, you see the same protein and its coding mRNA across different conditions. This is panel A. So in panel A, you basically ask if you change the transcript across different conditions, how is the protein responding? And the right-hand side, you have different proteins and they're coding mRNAs in the same condition. So here's only one condition or a steady state situation. And then you ask across the different genes and proteins, how much does how much do the protein levels depend on the mRNA levels? So the right-hand side is more static, and it's across different conditions. Across different genes, whereas on the left-hand side in panel A, you have different conditions in the same gene. And if you know a bit the literature, there are many papers where there's some kind of argument going on, how much should protein levels depend on RNA levels, and this, of course, very much depends on which of these two correlations you're really looking at. And so this is what I said. The left-hand correlation shows you the change of transcript in relation to the change of the protein, just to emphasize this again. And then on the right-hand side, how much do absolute protein levels depend on their coding mRNAs within one sample, typically. So how strong are these correlations that you now should expect if you do the experiment yourself, or if you read a paper and they are reporting a correlation? What should you expect? For the right-hand type of analysis, where you look across genes, you typically get a correlation between 0.4 and 0.9. And when I say correlation, this could be classical Pearson correlation. It can also be the Spearman-Rank correlation. Spearman-Rank correlation has the advantage that it's independent of the type of relationship between protein and RNA. There doesn't have to be a linear relationship, but typically they're both in the same ballpark. But of course, it always depends on the species. It depends on the cell type. It depends on, you know, how precise are your measurements, and so on and so on. So there are many factors that influence this correlation. For the left-hand side, so same protein across different conditions, this is much more complicated. This can be anything between 0 and 1. Seriously, yeah, because if you imagine a situation where you have different conditions where there are very small or basically no changes of the transcript and the protein across the conditions, then any variation that you have is random, so you expect a 0 correlation. Whereas if you have very drastic changes of transcripts and proteins, then it's also much easier to detect a correlation between the protein and the RNA across the conditions. So I don't think you can make any statement about, you know, what you should expect in this situation here. Now here's an example of a study just to exemplify the importance to distinguish these different types of correlations. So this was a paper where human proteins have been measured in different tissues, and then they did a number of different analyses, and one of the analyses they did is they compared the protein levels in these tissues to transcript levels from the same tissues. But this is also important, the transcript data was not from the same samples. This is, of course, important. That makes a difference. So in that case, you actually don't expect a correlation that's as nice as if you had protein and RNA from the same sample. But I think this is trivial. Now, what the authors did is they did the classical correlation analysis, and they didn't get very excitingly high correlations. But then they noticed that the ratio of the protein to the RNA for each protein-RNA pair was very constant across the different tissues. This is what you see on the top left corner. You see the different tissues, and then you see, for some example, proteins. Each protein is one color here. You see the ratio protein to RNA, and you can see that this is fairly constant across the different tissues. And now just focus on the bottom right panel. In this panel, they now use these ratios, the protein-RNA ratios, to predict the protein level in a given tissue using the respective mRNA. So now they assume that the protein-RNA ratio is constant, so this is not tissue-dependent. So the only tissue-dependent information here is the transcript, and using that they predict the protein level, and now they get a very high correlation of up to 0.91. So it was in the range of 0.8 to 0.9 what they had here in these correlations. So that's very impressive. But the question is now, what would happen if you now would use the wrong mRNA? So now if you would use the mRNA from a different tissue, you should expect that in this case you would get a much worse correlation because you're not using the right mRNA. So then another group did an analysis that goes in this direction. It's not exactly the same, but it's the same idea. So now this was published this year actually in Nature. So you see the correlation of the protein to mRNA in the left box plot for the different tissues. So here each of the black dots is the correlation of protein to RNA in one tissue. And you see that this is the Wilhelm et al. mRNA. In this case, you see that the correlations are around 0.4. And then on the right-hand side, you see the predictions using these protein-RNA ratios. And this is just repeating what I just said. These correlations are in the range of 0.8 to 0.9, so they are much higher. So using these protein-RNA ratios, you get a much better correlation. And in this paper, the author called these ratios the translation rates, although I think that this ratio is also affected by the protein half-life or the turnover of proteins, so I think it's really summing more than just the translation rate, but that's the term they used. So now in the team of Fortelny et al., they did the following trick or test. They, instead of using mRNA data that was tissue-specific, they just used the median of the given mRNA for a certain gene across the tissues. So now they essentially average the transcript levels across the tissues, and then they use this average transcript level with the fixed protein-RNA ratio to predict the protein levels tissue specifically, and surprisingly they get a correlation that is almost as good as if they would use tissue-specific mRNA. Now this is suggesting that for this type of correlation, the tissue-specific mRNA is not really contributing much. And then they went even more crazy, if you like. So now they used random transcript levels. So they randomized the relationship between protein and transcript and repeated this analysis. And they again get a correlation that is very close to the one using the true transcript levels for the specific tissue. Now what's behind this? Well, the problem here is really this distinction between these different types of correlations. And here's a simulation that they did just to essentially visualize this problem. So here on the left-hand side, you see the protein levels for different 3 different genes, and then the respective mRNA levels. And you see that the blue gene C, has on average relatively high protein levels, and the red gene A, has relatively low protein levels. And then you have, in addition to these, if you like, static differences between the genes, you have variation across the different tissues, and these are the different dots that you can see. And then they did the regression in the simulated data, and you can see that there's a dependency of the protein level on the mRNA level. So the higher the mRNA, the higher the protein. It's not a strong correlation, but there's some dependency. On the right-hand side, you see a very similar situation, the same genes A, B, C, but now in this case, they assume that there's no dependency of the protein on the mRNA in terms of variation across the tissues. Yeah? So across the tissues, there are different mRNA levels, but there's no correlation anymore. And so these gray lines are all horizontal. In both panels, however, the protein to mRNA ratio would be very predictive for the average protein levels. So you would assume that the blue protein is usually very high and the red protein is usually very low. And if you would then just correlate the predicted protein levels to the observed protein levels across the different genes, you would get a pretty good correlation in both cases. But in the right-hand case, the mRNA doesn't really matter. So this is emphasizing again this point that you have to ask yourself, what is it that's interests me? Is it the differences between the different proteins and genes, or is it the variation of the same protein across different conditions like different tissues? So here they did the reverse analysis. So now in this case, they look at the variation across the different tissues for the same proteins, and then they did the protein to RNA correlation. And what you can see here is the distribution of these protein to RNA correlations for all the proteins where the data was available. And you see that in this case, the average correlation or median correlation is just 0.2, so it's much lower. So the true contribution of the mRNA change in this data set is relatively low for explaining the protein change across the tissues. But this is not a bad result. That's very important. I want to emphasize that. It's not saying that the data is bad or anything. We are really looking at a different question in this case, and it's also important, again, to keep in mind that the transcript data was not coming from the same samples as the protein data. I think if you would repeat this analysis with transcript and protein data from the same tissues, also this type of correlation would be much higher. Okay, so now we come to the biology and also some technical issues. What really determines protein levels? So you all know the central dogma. In order to make a protein, you make the mRNA is translated, you get the protein, and then the protein at some point is turned over, it's degraded, and that's it basically. But of course the reality is much more complex because each of these different molecular concentrations that you have, the mRNA concentration, protein concentration, and all these processes are affected by many additional processes that are controlling them. So, for instance, the mRNA levels are also determined by the stability of the mRNA and also the effective mRNA. So the mRNA that is actually available for translation also depends, of course, on the localization of the mRNA. This is particularly important in large cells, like in neurons, for instance. makes a big difference where in the cell the mRNA is for the extent to which it's going to be translated. Then translation, of course, depends on many other things. It depends on the structure of the mRNA, the folding of the mRNA. It depends on proteins that are regulating translation. They might bind to the mRNA. It may be affected by microRNAs that could block translation. It depends a lot on the codon usage, so the composition of the protein sequence. And of course, it depends also on the availability of the charged tRNAs. Also, that might change in stress conditions. In fact, I know of a study where it has been shown that changes in the tRNA pool in cancer can actually trigger the translation of oncogenes. So also that is definitely very important for understanding the protein levels. Then protein turnover is affected by, of course, the proteasome and to what extent the proteasome is actually available, because also the proteasome might be blocked or clogged in a way. Then autophagy, you can have more or less autophagy. The stoichiometry of protein complexes is very important. I will talk about this later. And then, of course, protein folding matters a lot here. If you have, for instance, protein aggregation, as in Alzheimer's or Parkinson's disease, etc., these protein aggregates are very stable, or they can be at least very, very stable. So that would have a dramatic impact on the half-life of the proteins, and possibly many other processes that I didn't mention here. And then, of course, the protein can also be exported out of the cell, so then it's gone. It's not together with the mRNA anymore where it has been translated, or the protein might be imported into the cell, and in this case you also get a decoupling of the protein and its coding mRNA, which is important, especially if you have very high resolution measurements, single cell measurements, for instance. Then the protein that you see in the cell may not come from the cell, and the mRNA from which it got translated would also not be in that cell, and then you get also, of course, not a very high correlation. I'm going to say also a bit more about the importance of the resolution later. So these are just the biological processes that are important for the protein to mRNA relationship. What about technical issues? Of course, there are many. These are, again, just examples. Post-translational modifications, for example, might change the peptides that I can detect with the mass spec. So the peptide might be there, but because it's phosphorylated, you don't see it basically anymore in the mass spec. You may have incomplete proteins that, you know, the translation was blocked, or they have been started to be degraded, but you still see certain protein fragments and peptides. Then splicing is still a big issue. How do you distinguish different splice isoforms in proteomics data? But this would be important if you want to relate the protein that you measure to the splicing isoform of its coding transcript. And then, of course, some proteins are harder to measure than others, like membrane proteins, and then you might have biases also there, which are purely technical and not really biological. This was proteomics, but at the transcriptomics side, of course, you also have issues. In a cell, you always have immature nascent RNA in the nucleus, which is not translated. If you measure the whole cell, you will still measure the nascent transcripts, even though they are not available for translation. Splicing also is an issue here with the transcriptome data, and transcript stability, of course, also matters in this case. So here, transcript stability, this triggers basically the next question. We have to look at the kinetics that's going on here. So here's a figure to visualize the problem that we have. The translation creates, or the maturation of the mRNA and then the translation creates basically a delay between the transcriptional response and the upregulation of the protein. So if you imagine a situation where you have some stimulus that triggers the transcription of a certain gene, then first, of course, also only the mRNA goes up, and then it will take a while until you see the upregulation of also the protein. Now, if the mRNA is relatively unstable, which is usually the case, and the protein is very stable compared to the mRNA, then you might get a situation like this one, where the protein level goes to its maximum long after the mRNA has reached its maximum. So if you would then measure the protein levels at a later time point, you wouldn't see the mRNA anymore. The mRNA that has synthesized the protein is already gone. And this could also, of course, create a decoupling, if you like, between protein and mRNA. So if you measure the correlation at this time point, you might see a poor correlation, even though the protein level actually completely depends on the mRNA in this example. The kinetics also plays a role. If you look at single cells, we think that transcription often works in bursts. So in a single cell, there are certain time points where you're heavily transcribing a certain gene, but then there are maybe other times where the transcription of this gene is silenced. And then you wouldn't see these transcription peaks if you would average across millions or billions of cells, what you usually do if you do bulk RNA sequencing. But as soon as you do single cell sequencing, you can see these transcription bursts. Now, if you look at the protein level, the protein might be much more stable than the transcript. So again, the protein levels are, if you like, kinetically decoupled from the transcript. And now in the single cell situation, if you were able to quantify protein and RNA from the same cell, you might not see a good correlation because the transcription spike has been before you were quantifying the protein, so you don't see the RNA anymore. The similar situation applies to cell cycle dependent genes. Of course, transcripts are cycling in these proliferating cells, and the proteins are also, of course, cycling. But again, you have this issue of the delay. So if you do single cell measurements, you may not see the protein and the transcript at the same time in the same cell. Whereas if you average over millions and billions of cells, you would not see this, or you would not have this problem. Yeah? So this basically leads to an important conclusion here. As the resolution of our measurements is increasing, we might actually see a reduction in the protein-RNA correlation because we are averaging less, spatially and also in terms of time. I was already mentioning the transport of proteins between cells. If you have something like that, if you do this at a single cell level, the spatial resolution is so high that you may actually get a poorer correlation as opposed to measuring, let's say, a whole tissue. So this is one important point to keep in mind. There are many studies that have investigated the degree to what transcripts contribute to mRNA levels. I would like to look at one study more in detail because I like it a lot. I think this is a really, really nice paper. It was published in 2015 by Aviv Regev and her team. In this study, they looked at LPS-stimulated dendritic cells. So important here, these cells are basically, if you like, designed to respond to LPS. So that what you see as a response is a very initial response of these cells. And then they measured very extensively and in detail protein levels, also protein level changes through pulsed SILAC, but also RNA synthesis and the turnover of proteins. And then they combined all of this data in models. They created models for each transcript protein pair to essentially model or simulate, if you like, the production and degradation of the protein as a function of transcript, but also as a function of the other processes. And now what you can see here in these sorry, a few more points that are important from this paper. It's important to distinguish the steady state situation, essentially before the treatment with the LPS, versus the dynamic changes, so looking at fold changes after treatment, before treatment. So they also make this point that this is very important to distinguish. And it's also, that's another conclusion, very important to distinguish relative changes versus absolute changes. Now I'm trying to just emphasize this with this one figure that I have pulled out of the publication. So on the left-hand side in panels A and B, you see the contributions of transcriptions or transcript levels, translation and degradation to protein levels before the treatment. So this is the steady state situation that you have. And on the right-hand side, panels C and D, you see the changes after the LPS stimulation. On the left-hand side, you have two panels because they use different ways of quantifying translation. I'm not going into details here, but the conclusion is basically the same. You have roughly 70% contribution from transcripts, 20 to 30% translation and about 10% degradation that are contributing. Now, after the stimulation, the relative fold changes are almost completely determined by transcript level changes. So for most of the proteins, the change of these protein levels is completely explained by the change of their own transcripts. And then there's very little contribution through the regulation of translation and degradation. If, however, you look at the absolute changes, so if you count the number of molecules that you have in a cell, the picture is very different. So now you have only 40% contribution from transcripts. And the explanation that the authors had in this case was that for very highly abundant proteins, even very small changes in the, for instance, the translation rate would have a huge impact on the total number of proteins that are created, the number of protein molecules of the species, and this would then contribute to changes in absolute numbers of proteins. And I think it is also emerging from the literature that the total number of proteins in a cell is relatively fixed, unless the cell is growing, of course. So that means if you are putting more resources into one protein species, you necessarily have to reduce the concentrations of other proteins in that cell. Okay. Now the slides are not responding. Here we go. So here's an example for the kinetics from the same paper. Here they looked at 3 different proteins or protein transcript pairs. They quantify the transcript changes as a function of time. This is the top row of these panels. Then you see the changes in translation. Below this is derived from the modeling that I mentioned before. Then you have the degradation, the change in degradation in the third row, and the change in total protein in the bottom row. And you can learn a lot of things from these examples. One example is, for instance, you clearly see the delay of protein level change compared to protein transcript level change. So for instance, for TRAFD1, you can see that there's the peak of the transcript already at 6 hours, whereas the proteins continuously increase towards the end of the whole time course. And you can see similar things for the other proteins. Interesting is also REL-A, where the transcript level under the LPS state treatment goes up, then it goes down, whereas the protein level remains relatively constant. So in this case, there's an increase in the translation, but also in the degradation of the protein at the same time, and that would explain why the protein remains stable. I don't know why this protein is regulated this way, but that's what they observed here. But it shows you how important it is to really consider the kinetics. Okay, so now the next chapter is when is post-transcriptional regulation really relevant? So when is it not enough to just look at the transcripts? First of all, why is it attractive to use mRNAs as a proxy for protein levels? First of all, mRNA is relatively easy to measure, even though it's unstable. The technology of RNA sequencing is pretty mature. It gives you very easily a genome-wide measure. You get genome-wide measures of transcript levels. As opposed to that, many people think that proteomics is more noisy or more difficult to do than transcriptomics. And with proteomics, it's also difficult to really completely cover the whole proteome. This usually requires big efforts, especially in mammals. Whereas, as I said already, with transcriptomics, you get very easily the coverage of the whole genome. And then, of course, the question is, does it even matter? Does it matter that I take mRNA as a proxy for protein levels? I mean, as you've seen from the paper of Jovanovic, mRNA is contributing most to the protein levels. So I think it is important to just know or be aware of situations where protein levels might be largely determined by transcript levels, so that you consider this in your work. I came up with basically a rule of thumb. When is it? When a protein level is mostly determined by transcripts, and when is post-transcriptional regulation very important. And I think the rule is basically if you have a predictable change, and I will say what that means, then the protein level is mostly determined by transcript changes. And if you have something unpredictable, then the cell will utilize more post-transcriptional regulation. So predictable could be, for instance, the development of an organism, the development of an embryo. In this case, these are Drosophila embryos. This is a very deterministic system, a very deterministic change in transcription in different cells and cell types. This is fully programmed in the genome, and basically you can always express the transcript just before you need the protein, and the system kind of knows which protein is needed next. And so this is completely done via transcription. Another example would be the cell cycle. That's similar. So if the cell enters the cell cycle and it goes into S phase, it's clear which proteins are needed to go through S phase. So the cell can transcribe the genes that are needed in S phase just before S phase starts. Then it makes the proteins so that all the proteins are ready when the cell enters S phase. This is also just an example. So these are all processes that are predictable and very deterministic. That's different with stress situations. So if you stress the cell, then this is an external stimulus, and the cell may not be prepared for responding to this, or there may not be enough time to just transcribe the genes that are needed and then translate them. This might take too long. So in this case, that's an example where post-transcriptional regulation is very important. I think the nicest example that I know of is the regulation of GCN4 in budding yeast. And this is a protein that is a transcription factor that's responding to a starvation. And in this case, the transcript of GCN4 is always present in the cell. It's always transcribed. But then if there comes the stress stimulus, so if there's the stress signal, then the translation is turned on. So now you make the protein. And this allows the cell to produce the protein, in this case the transcription factor, very fast, because it doesn't have to transcribe the gene first. But it's also not wasting amino acids into a protein that is not needed. You only make the protein when you actually need it. We call this process translation on demand. We, more than 10 years ago, did a more systematic analysis for finding such proteins that are regulated this way. And in fact, our analysis revealed that mostly transcription factors are regulated through translation on demand. And in the meantime, this has also been confirmed by others, including, by the way, the Jovanovic study. They saw the same. The small number of proteins that were regulated translationally were enriched for transcription factors. And if you think about it, this makes a lot of sense. For instance, receptors that sense a stress signal, they have to be always present. Otherwise, you cannot sense the stress. So these receptors cannot be regulated through translation on demand. If you want then the transcriptional response of the cell, you need the transcription factor. So the transcription factor is somewhere in between the receptors that are sensing the stress and the genes that are regulated. So these transcription factors are then regulated through translation on demand, which speeds up the response. Translation itself very often follows the changes in transcription. So I just described to you the exceptions from this rule, but across genes, across many genes, mostly translation follows transcription, which, if you think about it, makes a lot of sense, because why would the cell upregulate a transcript if then it would, for instance, downregulate the translation? That doesn't make much sense. Of course, there are always exceptions, but this is the general rule. Here's just one example study. There are many. There are more citations in the review. So in this case, the team around Leonid Krugliak and Jonathan Weissman, they quantified translation rates and transcript levels in different yeast strains. And what you're looking at here are the differences between the yeast strains. And on the x-axis, you see the change or the differences in mRNA levels between the strains. And on the y-axis, you see the differences in the translation rates. And you see this is highly correlated. So this is just one example. So that means that the translation is kind of amplifying effects of transcription in both directions. Yeah, they can both go up or both go down. Another very important process that contributes to protein levels, and that actually explains to a large extent differences between protein and mRNA levels, is what we call protein level buffering. So what do we mean by this? Here you have in this figure that you see, you have the same protein across different conditions, and in these different conditions or samples, you have variation of the mRNA, but the protein is relatively stable. So this basically says that the cell needs to control the protein because the protein is doing the work. So even if there's some kind of unwanted variation or fluctuation of the transcript, it still has to stabilize the protein, because otherwise you might, for instance, break the stoichiometry of certain metabolic processes or of protein complexes. And as it turns out, protein complex stoichiometry explains to a large extent this, what we call protein level buffering. So here's one example study that was published recently about cancer. So in cancers, you very often have variation of gene copy numbers, copy number alterations, CNAs. And you would expect, of course, that if you have an increase in the copy number, you get an increase in the transcript, you get an increase of the protein. If you have a deletion, you expect a reduction in the transcription, a reduction of the protein. And now what you see here is the correlation of the protein to the copy number change on the y-axis, and the correlation of the transcript to the copy number change on the x-axis. And you see this big gray cloud in the middle, which is basically saying that if the transcript correlates with the copy number change, the protein will also correlate with the copy number change. Then you're in the upper right corner. If the transcript doesn't correlate with the copy number change, the protein also doesn't correlate with the copy number change, then you are at the bottom left. And then you have this big red cloud in the bottom right, and these are genes where the transcripts are highly correlated with the copy number change, but the proteins are not as highly correlated with the copy number change as you would expect. So these are buffered proteins. In this case, the protein doesn't follow the copy number change, less than what you would expect given the transcript response. And then the authors did an additional analysis that shows that this is largely due to proteins that are parts of protein complexes. The issue is that if you have a protein complex consisting of, for example, of 3 proteins, you need all 3 proteins so that you get a functional complex. If one of them is missing, you have a complex that is dysfunctional, and then the cell will degrade the dysfunctional protein complex. And that's what you see here. You see that the red dots are the proteins that are buffered. These proteins are enriched in functions that are in proteins that are parts of complexes, protein complexes. And the next plot shows this in a different way. So here you have the correlation between the different protein pairs, and if you take random protein pairs, this is the upper distribution that you see, the blue distribution. These random protein pairs are basically uncorrelated, so the correlation is around 0, the distribution is around 0. Random transcript pairs are also uncorrelated, you get a distribution around 0. But if you take protein pairs that are part of the same complex, these protein pairs are highly correlated. That's the blue distribution in the middle. Transcripts are also correlated. That's the red distribution that's shifted to the right, but not as much as the proteins. And this is important. So the proteins are more controlled than the transcripts. And that's, again, emphasizing this buffering. And then the bottom is functional relationships, but I'm skipping this due to time. Here's an example from our own work. Coming to the same conclusion, but this is not cancer. Now we're looking at trisomy 21. Here you have data from 2 people. One person has trisomy 21, the other one does not. And then we quantify transcripts and proteins. And here you're looking at the distribution of transcript levels from genes encoded on chromosome 21 on the left, and all the other genes on the right. And you see what you expect. Transcripts from chromosome 21 are upregulated in the trisomy 21, and the rest is basically random around 0. This is what you would expect due to the increased copy number of the chromosome. Now, if you look at the proteins, the situation is very different. If you look at proteins that are part of protein complexes, so the pink ones, they are not responding to the transcript change. only proteins that are outside of protein complexes are responding. They are also upregulated, whereas those that are in a protein complex are degraded. This is what you see on the right-hand side. Their degradation rates are higher, and that's most likely because you create incomplete protein complexes which then get degraded. There are multiple other papers showing this phenomenon. I'm also referring you again to the review where you can find more citations. So now, very last aspect, and really brief, what else can you do with these protein RNA correlations other than just trying to understand the biology? And we think you can also use it for some kind of quality control or method development. Here's an example where we used mRNA as an independent external reference because we had different ways of quantifying proteins by summarizing peptide levels from proteomics data, and we didn't really know which way of summarizing is the best. And because if you use other proteomics data, you run basically into the problem that this other proteomics data might be analyzed in a different way, and then you cannot really compare your analysis to other proteomics data. So instead of using other proteomics data as a reference for deciding which method is the best, we were using mRNA levels. So we correlated the protein levels that we derived from the peptides with their coding mRNAs. In many samples, each of these creates a distribution. So the vertical box plots here are the distributions of correlations that you get, and then each box plot is one method, and then you see there are some differences. So there are some methods that have a lower correlation than other methods. So we could exclude those methods, for instance, whereas other methods are maybe equally good. So this would be a conclusion from this type of analysis. Of course, you have to be careful here because you may have to make sure that a better correlation between protein and RNA is not just due to some bias, because, for example, you select preferentially highly expressed transcripts, and then, of course, you also have better protein levels, something like this. So there are possible caveats here. We discussed those caveats in a paper that is cited at the bottom, Gatto et al. So if you want to read more about this, you can read this paper. Okay, here are references that you can read if you like. On the left are all these articles that I've cited in this presentation. Many, many more, of course, in the review that we wrote, but there are also many other reviews which are good. The reviews that are on the right, this is really just a subjective selection, and there are many more which are also very good. Yeah, I'm sure you could also find more of those. So basically, to just maybe conclude, protein levels are mostly determined by transcript levels, which is probably what you expect. I think it is established that transcription is the major process by which cells control the abundance of proteins, but there are these other processes, and they are important. They are important for certain proteins, and they are important in certain situations. So I think you have to be aware of this difference. And I think there are enough examples that also show that just using mRNA always as a proxy for protein levels is maybe not a good idea. So then I'm done. I thank you for your attention, and yeah, I'm acknowledging here only Yansheng Liu and Rudy Ebersol, who wrote this review with me. Of course, many other people have contributed to the work that I've cited. I thank Oliver again for organizing this webinar, and I thank you for your attention. And yeah, I hope you have some questions now, and I'm happy to answer those.
So thank you very much, Andreas. So I learned a lot, and I have now much more to discuss in the future with our customers. So as we are collecting the questions here, so they're coming in, I already have here some. So you mentioned this example of these transcription factors that were less regulated on the transcript level. So is it in general true that lower abundant proteins are less regulated by transcript level and higher abundance more, or is this? something?
No, I don't. First of all, I don't think you can conclude this. I don't think the level of post-transcriptional regulation, if I may simplify it like this, is dependent on the protein level. In fact, I think the Jovanovic paper is telling the opposite. So their conclusion is that the very high abundant proteins are what they call the housekeeping genes. They are the total amounts are highly dependent on the translation rates and the turnover, because if you increase the translation of a very abundant transcript just a little bit, this has a dramatic change on the total number of proteins that is created. And even for the transcription factors, it's also important to make this maybe more clear. If you have transcription factors that are, for instance, important in development, cell differentiation and so on, they are also mostly controlled via transcription. What I was referring to was more the stress response. So in fact, I think there are very few genes that are really regulated via translation on demand, but those are enriched for transcription factors.
Okay. Good, and then we have a question here regarding single-cell proteomics and single-cell RNA-seq. That is certainly becoming more fashionable, let's say. So do you expect to see a higher correlation then in between single-cell proteomics and single-cell RNA-seq data, or the opposite?
Yeah, of course it depends again on the situation, but at least for certain genes, I would expect a lower correlation. So if you have a very dynamic transcript that's unstable, but you have maybe a stable protein, then the protein and the transcript in the same cell they are basically decoupled, and then I would expect a lower concentration. If, on the other hand, you have let's say a transcript that is continuously transcribed at the same level, and then you make the protein, and the protein is also very stable, so if you have a more static situation, then the protein differences between different cells will be mostly dependent on the differences of the transcripts, and then you would get, for these proteins, you would get a maybe very good correlation or very high correlation at the single cell level. So the answer is it depends.
Okay. And then I think we have this protein buffering or protein level buffering aspect. So generally, is it fair to say that you expect more noise on the RNA level versus the protein level? Or again, is this?
Yeah, that's a very interesting question. So there have been papers that I couldn't cite here, but they are cited in the review by Jonathan Pritchard and Joaf Gilad, where they show, I think, very nicely that protein levels are under stronger evolutionary selection than transcript levels. So this would confirm what you were just suggesting across species. So across species, you can have some variation of transcripts that is essentially evolutionarily neutral if the cell is able to stabilize the protein, for instance, through degradation or other processes. So in this case, you would actually see more variation of the transcript levels than for the protein levels. We also have some very recent analysis that I couldn't show because it's really too premature, that is also suggesting that proteins are evolutionarily more controlled than transcripts using other types of data. So I think that's something that I believe, and that's the case. Making a more general statement, you know, if you have, I don't know, different cells or cell types, that you have more transcript variation than protein variation, I think such a statement is very difficult to make because, again, it depends really on the specific situation. But at least for this evolutionary scenario that I was describing, I think that's the case, yes.
Okay, and then we have an interesting question here. Again, probably answered it depends, but still to get a feeling for this. So which post-translational regulation would you say overall is more relevant for protein level buffering, the translational or the transcriptional level?
For protein level buffering, that's the degradation of the protein. It's really the turnover. This is, I think, pretty clear because there are now several studies that show that the translation usually follows the transcription. So if transcription goes up, translation goes up as well. That means if you have noisy transcription, so if you have an upregulation of a gene that is actually not really beneficial for the cell, the translation will follow that. So the translation is not going to buffer the transcription in this case. So the buffering is really happening at the level of protein turnover. And that is mostly due to the degradation of incomplete protein complexes. Because if you make a protein that's not really needed, it will not be incorporated in its protein complex, and then this will be degraded. So this is the current thinking. You know, there's no proof, but I think this is the explanation that is kind of emerging from the literature.
Okay, good. And then here, probably a hard question, also something that is interesting at the moment, circulating messenger RNA, different type of messenger RNA. How does this correlate with blood protein quantity at all?
With blood protein, I don't know. Yeah, I mean, first of all, I think it's not really clear what is the function of circular RNAs. There have been some suggestions, and for example, Nikolaus Rajewski has also shown for some example circles what they actually do. My impression is that still many circular RNAs are just errors of splicing, so they are not necessarily functional or they don't necessarily have intended functions. So if you have an excess of circular RNA formation that is just due to erroneous splicing, this would then reduce, as a consequence, it would reduce maybe the protein amount. But this is
actually, I think I misspelled that. I'm sorry, it's circulating messenger RNA.
Circulating, okay, circulating in the blood and then the protein. Okay, okay. Circulating RNAs can, of course, be from very different tissues, and the proteins could be from the same tissues where the circulating RNAs come from, but they don't have to. So I think in this case, you really have to look in detail. Yeah, it depends on the specific transcript species and protein species. I think if you can use the circulating RNA as a marker, I think that's the background of the question. Can you use the transcript as a marker for the protein? That I think it's again dependent on the specific protein. If the protein is only produced in one cell type, if it's, for instance, very cancer specific, and you see the transcript, you see the protein as well, then it's likely that they both come from a tumor, I guess.
Okay, so we have actually many interesting questions here. So if at the end of the seminar we can't answer them all, we still try to put them on the website, for instance, where the seminar or where the webinar is available. But here's one: are there cell types in general? So you showed us different cell types at the beginning of the presentation that are better, where the correlation generally is high, versus other tissue types or cell types where it's generally lower.
I'm not aware of a study where this has been done. I mean, you could just look at the Wilhelm paper. They have done it, right? And then they see differences. You always see differences. I'm not sure how much of these differences are already understood. I think that's the more interesting question, right? So at least what I think is that if you see such differences, they're not necessarily just a reflection of, let's say, variation in the experimental quality. So you, because that could be another concern, right? That maybe the transcript or protein quantification was not done as carefully in one tissue as it was done in another tissue. We have not done such an analysis. What we have done is we have compared different yeast strains, and we see variation of this correlation in different yeast strains, and we have evidence suggesting that this is a real biological difference in the correlation. And I think the same will be true for different cell types. But I am not aware of a study where this has been systematically investigated and also interpreted, really.
Mm-hmm. All right, and here we have a question again that relates to the interesting data from the protein complexes, where you show that the messenger are upregulated for the protein complexes is then not reflected in the end on the protein level. So do you think that, if I understand the question correctly, that there's a time window for the changes where this is actually reflected on the protein level, so the protein would just disappear again and the cell wastes energy, or does the cell make sure that, say, proteins are not even generated and wasted to get the balance in the complex?
I don't think there have been really detailed studies of the mechanisms, but I think the consensus is that you make the protein and then the protein is degraded because it doesn't get incorporated into a protein complex. So you do waste energy, if you like. And this can be a problem for the cell because you might clog the proteasome system. If you make too many proteins that need to be degraded, then you may actually clog the proteasome. And this could then, for instance, trigger protein aggregation because protein aggregates are not degraded. You may know that in trisomy 21 there's a higher incidence of Alzheimer's, and so that would be one possible explanation, but at this stage that is speculation.
Okay, so I think with this question we close the session. So please feel free to further send us your questions to our account, for instance, the info@biognosis, and then we will try our best to answer them. I want to thank everybody for attending the meeting. Again, want to point to our next webinar from Rudy Ebersol next Tuesday. And especially thank you, Andreas, very much for presenting here. And I wish everybody a nice day. Thanks.